Hey guys, Assalamu alaikum everyone. I hope you all are doing well. Today we are going to start chapter number 3 of MDCAT Biology 2025, that is biological molecules. Okay. In the previous two lectures, we have completed acellular life and we have also completed bioenergetics. So if you haven't checked out those videos, they're available on the channel. Please go and check them out as well. Right? These slides are prepared in accordance with the Federal Punjab, Sindh and Balochistan board books. Okay, and along with the KPK books as well, and in accordance with the PMDC syllabus, as you can see over here, that is of 2025. Okay, so the first topic we have is define and classify biological molecules. We have to classify them, classify their importance. Then we have to explain the important properties of water. Then we have to discuss about carbohydrates, proteins, lipids, RNA, conjugated molecules, and then the structure of DNA, right? And the Watson and the Crick model. All right. So this is going to be a very lengthy chapter. I'm going to split it into two or three videos if this is not sufficient in a single video, right? But let us just start for now. What are biological molecules? Basically, these can be organic as well as inorganic. Now, what do you mean by organic? Organic means the presence of carbon, okay? The C looks so bad. It's like this. The presence of carbon, right? And in inorganic molecules, we don't have carbon, okay? And these are produced by living organisms. These are essential for production of their structure, function, and their regulation of the body cells. This is just a basic slide, just a defined purpose so that we can move on to the complex topics. These are made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now they are classified into inorganic molecules. They don't contain carbon and hydrogen bonds. Okay, the inorganic molecules, they don't contain carbon and hydrogen bonds, and the examples are water, mineral salts. Okay, all of these mineral salts. Why? Because they don't have the C and H bond. Okay, whether it is single, double, or triple, they don't have the bonds, right? Or gases, that is oxygen and carbon dioxide. Okay. Next, we have organic molecules. Organic molecules, they contain carbon and hydrogen skeletons, and these are large macromolecules. Examples are carbohydrate, protein, lipids, and nucleic acid, which are the basic food groups that we have. All of them are the same. Right now, based on the importance of the roles in cells, we have got carbohydrates. These carbohydrates are made up of carbon, hydrogen, and oxygen in the ratio 2:1. Okay, H2O. Just like in water, we have got two hydrogen and one oxygen, right? Similarly, over here as well, we have got hydrogen and oxygen in the ratio 2:1. And the basic unit is known as monosaccharide. What is mono? Mono means one. It means single. Okay, glucose and fructose, these are monosaccharides. Why? See, the formula of glucose is C6H12O6. It means what happens is that carbon and oxygen, they are in the ratio 1:1, whereas carbon to hydrogen ratio is 1:2, and hydrogen to oxygen ratio is same as that of water, that is 2:1. Okay, now the functions of carbon is that it is a source of energy in the form of glucose. Wait a second. Yeah. Yeah, it is a source of energy in the form of glucose. It is also used as a storage material, that is glycogen in especially in animals, and as starch in plants. And it is used for structure as well, to provide a structure and to provide support. For example, cellulose and chitin. There are these examples: monosaccharides, oligosaccharides, and polysaccharides. Right. So monosaccharide means one, di means two, oligo means more than 3, basically 3 to 10, and polysaccharide means greater than 10. Okay. Next, we have lipids. Okay, first, what we are gonna do is that we are gonna, have a little bit overview of all of the functional groups, and after that, we are gonna discuss them in immense detail as well. Okay, I have a 90-page presentation prepared, so let's just study. Okay, now what is the introduction of lipids? Lipids are basically hydrophobic. They are composed of carbon, hydrogen, oxygen. Sometimes they are composed of phosphorus and nitrogen. There are some simple lipids, for example, fats, oils, waxes. Compound lipids, compound means they are combined with another molecule. For example, phospholipids, they are combined with phosphorus plus a lipid. Combination is known as phospholipid. This is basically conjugated molecules. Glycolipids, glycogen plus lipids, lipoproteins, lipid plus protein, lipoprotein. Then we have derived lipids, which are basically, these are not simple. These are converted from one form to another, which are steroids and prostaglandins. We are going to study, and both of these are what? These are hormones. Okay, steroids are hormones, right? Functions are energy storage in the form of triglycerides, hormones like steroids, membranes, a structure, for example, phospholipids. Okay, as we know that the cell membrane is made up of phospholipids, right? Next, we move on to proteins. Proteins are polymers of amino acids and they are linked together by peptide bonds. Now, what are proteins made up of? Obviously, they are made up of carbon and hydrogen, right? Then we have oxygen, we have nitrogen, we have got sulfur as well, right? These are used for transport, for example, hemoglobin, for structure like collagen, antibodies, movement, hormones, which are in the form of insulin, right? We have got fibrous proteins, globular, and conjugated proteins. Conjugated means combined with other molecules as well. Globular, fibrous, and globular proteins, both of them are simple proteins, right? And please remember the examples that antibodies and enzymes, both of them are globular proteins. This is very important for your MCQ. And keratin and collagen, both of them are what? Both of them are parts of the fibrous proteins. Okay, please remember these main key points. Next, we move on to nucleic acid. Now, what is a nucleic acid? It is polymers of nucleotides. What is a nucleotide? It means sugar plus a phosphate backbone plus nitrogenous base. Now, what is a nitrogenous base? It consists of four parts: adenine, thymine, cytosine, and guanine. And if it is an RNA, it also contains uracil in the place of thymine. Okay. So the types are DNA and RNA. DNA, it basically used as a storage material. It stores genetic information, and RNA is used for the synthesis of proteins. Now the functions are: it acts as a genetic material, protein coding, and it is also used as energy currency in the form of ATP and GTP. Okay, based on function. These are basically, we are going to study this later. Based on function, these are the biological molecules. For structure, we have got collagen, cellulose. Energy, for energy, we have got glucose is the main molecule for energy. Then we have ATP, triglycerides in the form of fat. Informational molecules that basically contain our genes. These are DNA and RNA. Next, we have catalytic molecules in the form of enzymes, and regulatory molecules for hormones and cytokines. Now, biological molecules on the basis of bonding. So we are doing a classification of biological molecules first, right? On the basis of bonding, firstly, we're going to study about covalent bonds. Okay. So for covalent bonds, we have got basically covalent bond means that sharing of electrons is happening, right? In ionic bonds, an electron is transferred from one molecule to another, but in covalent bonding, there is a sharing of electrons, right? In carbohydrate, there is a sharing of electron between carbon and hydrogen and carbon and oxygen, which are basically known as glycosidic bonds. And in lipids, we have got covalent carbon-carbon bond. We can also have carbon-hydrogen or ester bond, which is carbon and oxygen. Please remember this, that esters are formed in lipids. Okay. In proteins, we have got peptide bonds, which is a covalent amide linkage made up of nitrogen, carbon, and hydrogen. Okay, and also disulfide bridges. What do you mean by disulfide? It means sulfate, two sulfur molecules with a single covalent single covalent bond between them, which means that they are sharing two electrons. Next, we have nucleic acid that have a phosphodiester covalent bond between a sugar phosphate backbone. What happens is that there are covalent bonds in the nitrogenous bases. Okay, adenine and thymine, right? These form a covalent bond. Then we have cytosine and guanine. These form a covalent bond, right? So when these form covalent bonds together, what happens is that they are classified in the form of covalent bonding, right? The properties are that these are strong bonds. These are very stable structures. Okay, but these are insoluble or they are partially soluble in water. Why? Because covalent molecules, these are not really soluble in water because they are sharing electrons, and they define the structural framework of the cell. All these carbohydrates, proteins, fats, nucleic acid, they help to define the structural framework of all the cells, right? These are strong bonds which have got lots of stable structure. right? Next we classify biological molecules as molecules with predominantly ionic bonds. Now, what is an ionic bond? Basically, it is electrostatic force of attraction between oppositely charged ions. For example, when I form NaCl, right? This is from your chemistry, not related to this chapter, but just a bit overview. It is Na positive and Cl negative, right? So these two ions are being attracted towards each other. What is the mechanism of attraction? It is basically the electrostatic force of attraction, because there are oppositely charged ions, right? So that is why the attraction occurs, right? It is seen in inorganic salts as well as in electrolytes. Okay, in the salts we have got NaCl, KCl, CaCl2, and magnesium sulfate. Basically, all of the electrolytes that are present in the body, these are being shown over here. Let me just erase. Yeah, and now the ions in the body fluids, for example, sodium, potassium, calcium, magnesium, all of these form the basis of solution in our body, right? So these are present in the body fluids. The properties are that they are easily soluble in water. Okay, covalent bonds, the compounds which have covalent bonds in between them, they are not soluble in water, but the compounds which have ionic bonds in between them, these are easily soluble in water. They dissociate into ions, they maintain the osmotic balance, nerve impulse conduction, muscle contraction, and pH buffering. So they help to maintain the osmotic balance. Why? Because they have got all of the inorganic salts, they have got electrolytes, they help in nervous impulse conduction, because nervous impulse conduction, it requires potassium and sodium, as you're gonna see later on in your chapter, right? It helps in muscle contraction as well, because it's all a game of ions, right? So wherever we have got ions, it helps in contraction, conduction, and in osmotic balance. Now, there are certain molecules which have mixed interactions, which have got both covalent bonds as well as ionic bonds. They have covalent bonds mainly for a backbone structure, but they have got ionic interactions by which they rely for stability, folding, as well as function. Okay. So first we have proteins. Proteins have got ionic bonds which have salt bridges between the charged amino acid side chains. Okay, whenever amino acids are joined to each other, basically linking of amino acids to one another is formed by ionic bond. And in between the structure of an amino acid, what is present in the amide linkage, we have got the covalent bond. Okay, in nucleic acid, when they interact with a magnesium and histones, they help to stabilize DNA. You just need to remember the examples, no need to see the structures. That when they make form with ions, for example magnesium, then they use ionic bonds. Okay, but when they have their own structure, then they form covalent bonds. And in cell membranes and phospholipids, we have got a covalent structure plus ionic interaction with other proteins or other ions. So a basic structure is made up of covalent bond, okay, the internal structure. But when this whole structure has to join with another structure, then what we use, then to join this structure, we use an ionic bond. So I hope you understand the difference. Next, we have got hydrogen bonding in the DNA. DNA is basically what? It is a double helix structure with two antiparallel strands. Okay, see, this is oops, wait a second. It just wait a second. Let me just yep. Let's leave it. Okay. So it's basically like this, a helix-like structure which has got two antiparallel strands, and these are held together by hydrogen bonds in between the complementary bases. Okay. So what happens over here is that adenine pairs with thymine with the help of two hydrogen bonds. Okay. See, adenine pairs with thymine with the help of two hydrogen bonds, and cytosine pairs with guanine with the help of three hydrogen bonds together. Okay, right? So adenine and thymine bonds, these are relatively weaker and these are easier to break, whereas cytosine and guanine bonds, these are stronger and they make this guanine and cytosine rich, and DNA is more stable. Okay, hydrogen bonding in the RNA. RNA is single-stranded, so hydrogen bonding is basically less extensive, right? But RNA can fold back on itself, just like hairpin loops or stem loop structures, right? In some of them, RNA exists as a double-stranded helix. So what happens in between this helix? Hydrogen bonding can occur in between this helix. And also there's intramolecular hydrogen bonding that is needed to join one molecule of RNA with another molecule of RNA. So when RNA and RNA are joining together, it forms hydrogen bonds. Okay. And also in DNA, when we have adenine and thymine, we have got two hydrogen hydrogen bond. When we have cytosine and guanine, we have got two hydrogen bonds, right? But in order to join them together, right? When, for example, this is adenine and this is thymine with two hydrogen bonds, then we have cytosine, then we have guanine. So the whole chain continues like this, right? But in order to join these two, these adenine and cytosine, then thymine and guanine, what are used? Covalent bonds are used. Okay, so don't get confused. Bonds between adenine and thymine and cytosine and guanine, these are hydrogen bonds. But in order to carry the whole chain, in order to support the whole chain, what are used? Which bonds are used? We have got covalent bonds. And if this DNA needs to combine with any other ion, any other ionic molecule, then only an ionic bond is used. Okay, so this is a major difference. Please don't ignore this, right? Base pairing in RNA. Adenine pairs with uracil instead of thymine. Okay, so in DNA we had thymine, but in RNA we have got uracil. Okay, and two hydrogen bonds are produced when adenine binds with uracil, right? And when guanine binds with cytosine, three hydrogen bonds are used as we studied earlier. Right now, the importance of biological molecules, these basically provide energy. Carbohydrates are the immediate source of energy. Lipids provide long-term energy and they provide more energy as well. Carbohydrate provide almost like 4 kilocalories and lipid provide 9 kilocalories. And proteins, they help in starvation. They provide energy during starvation. Next, we have structure, in which cellulose is an important structure of the cell wall of plants. Next, we have collagen and keratin in animals, which are basically forming the strong structures inside our body. For example, hair, they are made up of keratin, right? Why? Because these are very strong. Okay, next we have phospholipids, which help to form the cell membranes. Next we have the catalysis, that is enzymes. Enzymes are proteins which speed up the chemical reactions. Next we also have hormones, which is not mentioned over here, but you need to know this. Genetic information is stored in the form of DNA. RNA is used for protein synthesis, and ATP is used as energy currency. Okay, so it is actually mentioned, hormones are actually mentioned in the regulation, that it is used as insulin and steroids, which are very common hormones. Next, we move on to defense. Defense, we have got antibodies. Defense is basically the whole immune system of an organism, right? It produces antibodies and it also produces blood clotting proteins that help, that basically don't allow any infection to enter our body. Next, there is transport, which is in the form of hemoglobin, lipoproteins, and fats. They basically are the carrier proteins, channel proteins, and carrier proteins, which help to take molecules from one part to another. And hemoglobin, it basically is the carrier of oxygen. Hemostasis, it basically ions help to maintain osmotic balance, pH, nerve impulses, and muscular contraction, as we just studied earlier in ionic molecules. Okay, so we have done this part of our lecture. Wait a second. We are done with this. Define and classify biological molecules and discuss the importance of biological molecules. We are done with both of these. Now we move on to the biological importance of water. Let's read through. Describe biologically important properties of water: polarity, hydrolysis, specific heat, water as a solvent, reagent, density, cohesion, and ionization. We need to discuss all of these properties of water. And after that, we're going to move on to carbohydrates, proteins, and lipids, and further on. Okay, so let us start with water. Okay, water is a polar molecule. Now, what do you mean by polar molecule? It basically has got poles. Okay, what is the negative pole? Oxygen is the negative pole and hydrogen is the positive pole. Okay, why? Because oxygen is O2 negative and hydrogen is positive. Okay. Due to polarity, water forms hydrogen bonds with other molecules. This is polar, that is why it helps to form hydrogen bonds, right? And this helps to make water an excellent solvent and it allows interaction with the charged molecules. See, this is one molecule of oxygen and these are two molecules of hydrogen together with the water, right? Now this is what? This is negative, partially negative. This is partially, these are partially positive. So what happens is that these can attract other molecules to itself. Why? Because these are electrostatically charged. Okay, these are weaker than a totally ionic molecule, but these are, you know, a strong covalent molecule, right? So these attract other molecules towards itself, and it helps in hydrogen bonding. Why? Because all of these hydrogens, they combine and they form their own structure by which water can gain a lot of strength. And it makes water an excellent solvent. Why? Because it helps in dissolving, right? Polarity, it helps in dissolving because these negative and positive molecules, these can dissolve very easily with one another, and it allows interaction with other charged molecules, for example, ions, proteins, and DNA. Okay, so if the molecule is positively charged, still it will interact with this water molecule, and if the molecule is negatively charged, still it's going to interact with the water molecule. Okay, next we move on to hydrolysis. What is hydrolysis? Hydrolysis means breaking down of covalent bonds. Okay, what happens is that water, this is H2O, it breaks down into H, 2H positive and O2 negative, right? Now this helps in breaking other covalent bonds. When these two molecules are formed, 2H positive and O2 negative, what happens is that hydrolysis of ATP occurs and it is converted into ADP. So ATP can break down into ADP and it produces phosphate inorganic, plus it produces energy, and it is used in digestion of carbohydrates, proteins, and lipids. Okay, so what it does is that first it breaks down its own structure, then it attracts other covalent bonds towards itself, and it breaks down those covalent compounds as well, right? That is why it helps to speed up the process of digestion, right? Now, biological importance is that it is essential in catabolic reactions and general metabolism. Okay, what is catabolic reaction? Catabolic means break down. Okay, it means the breakdown of complex to simple. Okay, right? And what is anabolic? It means making from simple to complex molecule, right? Next, we move on to the specific heat capacity. Water can absorb or release large amount of heat without big temperature change. Okay, it can easily absorb heat, right? And its temperature does not change. That is why it is very stable. That is why our body is made up of 70% water. Why? Because it helps to make our body temperature stable, that is 37 degrees Celsius, and it also helps to create a stable aquatic environment for life, and it maintains a stable body temperature. Okay, why? Because it has a high specific heat. More of these reasons, why are they related chemically, you're going to see in chemistry. Okay, so please refer to your chemistry book as well. What is a solvent and reagent? It is basically used as a solvent. Why? Because it is polar, right? So it can easily dissolve into many molecules. It acts as the medium for biochemical reactions, and it helps in the transport of nutrients and waste. Okay, so it basically helps in digestion. Now this digestion can be physical as well as chemical. In our mouth we have got saliva has a constituent of water. In our stomach we have got we have got gastric acid, that is also made up of water. We have got mucus, secretions, enzymes, right? All of them contain some of the water as well, the liquid as well, right? So why does it contain it helps in digestion. Okay, it is a reactant in photosynthesis. It helps in hydrolysis and other reactions, and it helps to dissolve salt, sugar, gases, and it acts as a medium for reaction. Okay, why? Because it helps to dissolve everything. Water is denser than ice. Okay, density is that water is more dense than ice. Why? Because it is used as it is because of its hydrogen bonding structure. Okay, that it is just so strong that it becomes very dense as compared to ice. Ice forms an insulating layer protecting aquatic life in water. Okay, water is density, it supports aquatic organisms. So what happens is that this is an ice core which forms over the ponds and all the things, right? And this is water. Water is less dense. Water is more dense as compared to ice. So that is why it sinks and it is at the bottom, right? It is not at the top. At the top, we have got what? We have got ice. So the ice remains frozen, but it acts as an insulating layer and it acts as a supporting layer that helps to support these aquatic organisms which are surviving under the water. Okay. Next, we have cohesion and ionization. Okay, so what is cohesion? Cohesion and adhesion means that water molecules basically stick together. This leads to capillary action, that is xylem transport. So what happens is that xylem is a very sturdy sort of a structure in the stem that helps in the transport of water from the root to the leaf. Okay, what happens over here? These water molecules are sticking over here. They help to push other water molecules from the root up to the stem. So it basically acts as a support transport, and this is capillary action, and it also helps in the surface tension. More about surface tension you're going to study in chemistry, and more about capillary action as well. But for now, just understand that the water molecules, they stick together due to strong hydrogen bonds. So basically, the main characteristic of water, the jewel in the crown, is the hydrogen bond, by which water has got so many properties such as cohesion and adhesion, capillary action, surface tension, and then we have got ionization, that water can dissociate into H positive and OH negative ions, right? And it maintains pH balance, and it buffers biological system. Okay, so how does it maintain pH balance? Basically, it has got H positive, right, which is acidic, right? And also it has got OH negative, which is basic. So when these two combine, what happens? It is basically neutral. Okay, neutral, which is a pH of 7, right? That is why it acts as a buffer. That is why it acts as an intermediate medium. That is why it helps to maintain the pH in our body, because the pH of our body is mostly 7, except of some of the organs in the digestive system. Okay. Next, we move on to carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen. In this ratio, carbon is 1, hydrogen is 2, and oxygen is 1. Okay, the general formula is CnH2n, and N. Okay. Cn, for example, N is 1, so it is C1, then H would be multiplied by 2. Okay, so H2 and then O1. Okay, so hydrogen is always in a double ratio, and they are also known as saccharides or sugars. Okay, the scientific word for sugar is known as saccharide. Now these carbohydrates are classified, as we studied earlier: monosaccharides, di, oligo, and polysaccharides. These are the examples. Please take a screenshot of this slide and learn this by heart, because they ask you what are the monosaccharides, disaccharides, and polysaccharides. Right, so we have got glucose, fructose, and galactose in the main monosaccharides that have got one sugar molecule. Then di, we have got two: sucrose, lactose, and maltose. And oligo, we have got raffinose and styrose. And polysaccharide, we have got starch, glycogen, and cellulose. So we're going to study about these later on. First, we're going to study about their differences. Then we have monosaccharides, the properties of monosaccharides. Then we have the chemical structure and the isomerism of glucose. Okay, so it's actually a pretty long chapter. So I'm thinking to end the video right now, because my laptop isn't supporting so much of content right now. Okay, so we are gonna start from table 2.2 in the next video, inshallah, which is gonna be uploaded as part 2. Okay, if you don't understand anything, please let me know. And I'm ending this video over here. Inshallah, let's meet in part 2. Okay, take care. Allah Hafiz.